So next speaker is Jay Kormish from University of Manitoba in Winnipeg.
So thank you. I'd like to thank the organizers for inviting me to give a talk, and to the audience for not leaving to line up for supper early. So what I'm going to talk to you about today is some early day experiments that we've been working on in the lab concerning how the gland cells of the pharynx develop, in particular the unique morphogenic movements that they're using to make their cellular structure, and some of the signaling events that we're starting to find are involved in this process. So we, in the lab, study the pharynx, use the pharynx to study organogenesis. And we use this organ in that, even though it's a relatively simple structure, it has many diverse cell types that you might find in more complex organisms, such as neurons, muscles, secretory cells such as the gland cells, structural cells. And what I'm interested in is how these different cell types coordinate their movements during organogenesis. So how are they interacting with each other, and how do they accommodate each other's migratory patterns? So today I'm going to talk about briefly 3 signaling events that we think are occurring, and how we're using genetics to try and understand this process. So we are, just to give you an overview of what the gland cells are doing, is there are 5 gland cells within the developing pharynx. They all have their cell bodies located in the posterior part of the pharynx, in the terminal bulb. They have extensions which terminate at more anterior positions within the pharynx. They make a connection to the lumen and produce secretions that aid in digestion. We're most interested in this gland cell called the G1P cell, which is the dorsal cell, and it has an extension that extends almost the entire length of the worm. I've studied this because during my postdoctoral work, I found that it undergoes a unique process called retrograde extension to form its extension. This was first described by Shai Shaham's lab in dendrite formation. And this cell, in contrast to how most cells produce an extension with anterior grade extension, is born in proximity to where it will anchor its extension and then move through the pharynx, migrate through the pharynx, leaving this extension in its migratory wake. Initially, when I started looking at this, I thought they would have some unique molecular underpinnings to this process compared to, for instance, growth cone outgrowth. And so to start this, we first of all started looking at some live imaging studies in collaboration with Martin Sareko at the University of Alberta. And we started to look at this, at how it's migrating with respect to the muscle cells over time. And so what we found with the gland cells labeled in red and the apical junctions labeled in green to mark the muscle cell boundaries is that the gland cell initially abuts the PM4, PM5 border, and then about 400 minutes post-fertilization, it starts to migrate past the PM4, PM5, and 6 muscles to make their way to the posterior bulb. An interesting feature of this is the gland cell is in direct contact with the muscle cells during this point in time. And the muscles also will start to go elongation along the anterior posterior axis. And so that, I would imagine, would be inhibitory. The connections that the muscles need to make would be inhibitory to migration. So the cell needs to coordinate this with elongation, as well as the muscle cells accommodating this migration. So we started to look at potential regulatory mechanisms, and what we found were 3 receptors so far. 2 were AGL15, the single FGF receptor within worms, and INA1, one of the alpha integrins. And what we found in these mutant backgrounds is that the gland cell produced a phenotype where it over-migrated its its position within the terminal bulb and invaded the interring as well as the intestine, suggesting that these two molecules are probably providing some sort of stop cue. In addition, we found a positive cue for migration with it being CAM1, which is Aurora receptor, ROR2 receptor, a non-canonical Wnt receptor that can regulate the Wnt pathway as well as components of planar cell polarity. And what we found in these backgrounds, the mutant backgrounds for CAM1, is that that under-migration occurs, suggesting that it's providing some sort of positive cue for migration. So I needed to find a way to try and quantify this, and so what we've done is used the muscle position which the cell body interacts with at the time of hatching. And with this, we've been able to quantify the migratory defects, so that in the wild-type scenario, almost all of the cells associate with the PM7 muscle group, whereas in CAM1 mutant backgrounds, for instance, KS52, which has kinase domain deletion, as well as IVS36, which has Wnt binding domain deletion, that both of these alleles are required for this migratory event. This was interesting to us because so far a kinase-dependent function of CAM1 has been somewhat elusive. There is a phenotype in the vulval precursor cells in a sensitized background, but this particular defect, this penetrant defect, will allow us to hopefully more be able to investigate the kinase-dependent activity. And so with this, I tried to then determine what cell types they are acting within, and to do this, we used tissue-specific rescue experiments. In these experiments, we had a rescuing CAM1 construct, where black was were embryos that were positive for the construct, and red were embryos, sibling matched embryos that were missing the construct. And with this, we were able to find that expression within the gland cells alone, or the muscle cells that surround the gland cells, were not able to rescue expression. But when they were expressed in both cell types, with the KO7C11 promoter, or -4 promoter, that a robust rescue was seen. So this suggests that CAM1 is acting in a non-autonomous and autonomous way with regards to regulating the expression. So far, we've been trying to look at some of the downstream factors involved, and I've looked at most of the Wnt pathway as well as components of the planar cell polarity, and so far have not been able to identify downstream effectors. So a screen that I'm going to talk about at the end is hopefully going to help us identify what's involved in this process. And then with regards to FGF, we've been looking at how it's possibly producing this stop signal. Although I won't show it today, we did protein deletion studies that determined that the kinase domain is required. And using tissue-specific rescue experiments, again, we tried to identify what cell type it's working in. Initial expression studies suggested that it wasn't expressed in the pharynx. And so we attempted to, we thought it might be acting non-autonomously, and we used different promoters to try and address this. This was done by master student in my lab, Sinead Kim. And what we found is that none of these were able to rescue the migratory event. So we decided to look a little bit harder, that perhaps was a little bit more complex, and look more specifically at the different cell types within the pharynx. And with this, we found that that when we expressed CAM1 within the muscle cells as well as the entire pharynx, we did not see rescue. But when we expressed it within the gland cells alone, we saw, albeit not the same rescue we saw with the endogenous promoter, but a statistically significant rescue, suggesting that AGL15 is somehow acting within the cell itself, the migratory cell itself. Again, we were trying to find some downstream effectors. Obviously, FGF has many possibilities for this, depending upon cell context. We tried to address some of the major pathways to see if we could get a handle of what it's doing. And all those in bold, we tested for activity within this pathway and found that none of them are involved. So again, we're sort of hoping at this point that the genetic screen is going to help us out and identify how it's possibly acting. Finally, alpha-integrin was also seems to be involved in a stop cue. And again, it seems to be acting in a non-stereotypical way, in that when we look at INA1 null mutants, it gives a very penetrant over-migration defect. But when we looked at the other alpha-integrin and the beta-integrin, and in fact two separate null mutants with the beta-integrin, we found that they do not produce an over-migration defect. This suggests the possibility that these two are not acting together, and this is somewhat surprising in that alpha-integrin and beta-integrin are thought to be obligate heterodimers. There is some precedent for this from the Mormon lab that has found that muscle cell extensions seem to have an INA1 dependence without the function of PAT3. So they're certainly in the same place to interact, that both are expressed strongly within the developing pharynx. And so we kind of tried to test at least some very simple models with regards to what could possibly be going on and to try and confirm that INA1 is acting independently of beta-integrin. One was obvious maternal rescue. There's been some debate as to whether PAT3 persists at this stage of development. So we used RNAi to try and knock it down and hopefully reduce maternal function. Did not find a result. There was a truncated form of beta-catenin that's lacking the intracellular domain. We thought perhaps this might be interacting with the protein or with INA1, and we found that's not the case. And so we're trying to now identify perhaps I thought that INA1 may be acting to negatively regulate the PAT2, PAT3 integrin complex, so that in the INA1 mutant, you're gaining, actually creating a gain of function PAT3 function, and so we're trying to address that with double mutants right now. And so finally, we wanted to then look at what's potentially involved with regards to what integrin is interacting with, and it's been predicted to bind laminin within the extracellular matrix. And work done in Jim Priest's lab had shown that laminins are expressed in the pharynx during this time point of development in the basal lamina of the primordium. And so we, looking at first pass look at laminin mutants, for instance, LAM1, which is a core component of the laminins, we found that there is in fact an over-migration defect suggesting that somehow laminin is mediating the INA1 defect. We've also looked at other components, and so far LAM1 of the extracellular matrix, and so far LAM1 seems to be, laminin seems to be the best, our best guess so far. And so finally, we're hoping to use, so I have, unfortunately, quite a few more questions than answers right now with regards to how these pathways are working. And so we're hoping that the genetic screen that I'm looking at is going to help answer some questions. So far, it's been very productive at identifying over and under migration mutants. For instance, with CAM1, we've identified 24 alleles that phenocopy this, and so far we've found at least 7 that are non-allelic to CAM1. So we're hoping this will help us answer some questions. With that, I'd like to thank previous and current lab members, collaborators, and funding agencies. Thanks.
The over-migration phenotype looks, I think INA1 also causes that with distal tip cell. So,
which gene?
if I recall correctly, you can get over-migration of the DTC with loss of INA1.
Okay, yeah.
So I don't recall what else might give that phenotype with DTCs, or I was curious if you saw a strange gonad morphology with any of the mutants that you identified.
Well, INA1 is larval lethal, so I wasn't able to see. There is, although I haven't looked at it in detail, CAM1 does have a gonad defect, as far as I can, from just looking at the adult worms, yeah. But FGF, again, larval lethal, so it's been difficult to study later. Yeah, like a later knockdown or some sort of time-dependent knockdown. Yeah. Yep.
Can you help just orient me a little bit? So where would the laminin be that this cell is migrating on? So where is that matrix or basement membrane being set up?
Well, at least from work in Jim Priest's lab, it seems to be promoting apical basal polarity through expression on the basal lamina. And so, again, it's a little bit perplexing how that would interact with integrin if we were thinking that it's mediating the lateral adhesion between the cells. What I suspect is perhaps there's a possibility that maybe it's affecting the cell polarity and that somehow is affecting the general polarity of the cell, but perhaps there's expression between the cells that we're just not able to see, and so that's hopefully something we'll be able to look at. Yeah.
Are the cells that are over migrating faster, or do they just migrate longer time?
Yeah, so that was one thing that I wanted to try and figure out is its speed, right? They need to get to their point in development before the pharynx starts to elongate or it would be inhibitory. So far I can't exactly answer that. Although with CAM1, I have a suspicion that the cell sort of sits for a while, and I think it could be potentially a polarity defect. And so if you sit and you don't quite know your orientation and then develop your orientation due to redundant mechanisms, you might not get to where you need to go before elongation occurs. Yeah.
Do you know anything about what's holding the anterior tip of this gland cell in the pharynx? Is it DIF7 like?
No. So far, so through EM, at least later in development, there are electron-dense structures that are supporting the anchoring, but they have not been able to identify components of this electron-dense structure, so they're not apical junctions. So right now, what's anchoring this and creating that, the opening in the anchor is unknown. Yeah.
So if there are no further questions, I would like to thank Jay and thank everyone for this great session, including you for questions.